chromosomal disorder or any genetic disorder, how did it like get passed on to one person from another person? So that tracing is done by pedigree analysis. And in this analysis, we do like just like how in younger classes we used to draw a family tree. Same way here, this is a tree only, but of diseases. Now, in a pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. So we do so like how, like from where or from which person in the family the trait is being passed on from. Now, the pedigree analysis can be useful in detecting the diseases such as the Mendelian disorders. All of the Mendelian disorders that we had studied, those disorders can be detected depending upon the pedigree analysis. We can find out which trait is coming from which, you can say, generation or grandparents or parents. Now, for creating this pedigree chart or pedigree analysis, there are certain symbols that we use. And those symbols, the square is for an indicating an unaffected male, a male that is not affected with any disease. Then a circle is for unaffected female. And then this diamond shape is for the sex if it is unspecified. We do not know whether that is a male or a female. Now, once those shapes are filled in, then that represents the affected individuals, affected male, affected female, and affected sex specified person. Now, when in between the male and the female, there is a single line that represents mating. And when there is a double line that represents mating between relatives, meaning two related individuals. And then how do you study these charts? Here we see that, okay, mating, and here this is the first generation of children, and the order of birth is from the left to the right, meaning first this female was born and then the male was born. Now you're seeing here both the parents are unaffected, and same goes for the children, they are also unaffected. Now the parents with male child affected with disease, so the parents are normal but the male child is affected with disease, so it is depicted like this. And whenever in like diamond there is 5 written, so it represents that 5 unaffected offsprings are there. You do not know the gender of the offsprings, you do not know whether they are male or female, so you know that, okay, this couple had 5 children and 5 of them are unaffected. Now you do not know this couple with how many boys, how many girls are there. Now, by pedigree analysis, one can easily understand whether the trait is autosomal dominant or recessive, or whether the trait is sex-linked, just like hemophilia. So we can find out whether the trait that is in the human, that in the offspring, it is autosomal, or whether it is recessive. We can find out with the help of pedigree analysis. Now, in the case of recessive allele, characteristic condition can appear in the progeny of unaffected parents. Because why? Because it is a recessive form. And recessive form, once it gets into a homozygous condition, then it can be expressed easily, right? So that is what happens. Now, moving forward, two affected individuals cannot have an unaffected child. Now, two unaffected parents can have an unaffected child or an affected one, but two parents that are affected with a disease, a genetic disorder, they cannot have an offspring which is unaffected. The offspring has to be affected. Now, the types of inherited diseases are autosomal dominant, autosomal recessive, X-linked dominant, and X-linked recessive. These are the various types of diseases that we can study with the help of the pedigree analysis. Any doubts?
No, ma'am.
Pedigree analysis is clear to everyone? All right, students. Now, if it is clear, then you people can note it down, and then we'll start a new chapter. Here you can switch off your videos and note it down.
I'm done.
Everyone's done?
Yes, ma'am.
I'm done. I'm done. Ma'am, done. Ma'am, done. Ma'am, done.
Everyone's done? Everyone's done? Yes or no?
Yes, ma'am.
What about the others? Only 2 students are responding in the class. What about the others? Sarah, Sitika, Taneem, what happened to you all? No responses? Why? Yes, Sarah, just wait for a little while. We are like the chapter is completed only. Now we'll start the new chapter.
Ma'am, done.
All right, everyone's done?
Yes, ma'am.
Okay. All right. So now this chapter has been completed. We have actually completed the principle of inheritance and variations. And now we'll start a new chapter that is molecular basis of inheritance, the molecular biology. Now, everyone switch on your videos. Come on quickly. Okay, can you see my screen?
Yes,
Yes,
ma'am.
Okay. Where are the other students? Please switch on your videos. Don't make me ask you 10,000 times. Please don't do that. All right, so we are going to start this new chapter that is molecular basis of inheritance. Now, in this chapter, we are going to talk about the DNA, which is our genetic, like the genetic material, what are its properties, how it looks like. And then we are also going to find out how DNA only is the genetic material. Why not anything else? Why not there is like, there are like so many biomolecules, but why only DNA becomes our genetic material? So each of these things we are going to see. But before actually talking about the main things, there are certain things that you should remember. Now, you people, I don't know if you remember it from class 11th or not, but there are things about the bonds which are there. Now, first, initially, you might remember that the genetic material, it is of two types. The genetic material can be DNA or it can be RNA for some organisms. So either DNA or RNA. Now, who can tell me the full form of DNA?
Ma'am, deoxyribonucleic acid.
Very good. Deoxyribonucleic acid. And then what is the full form of RNA? Yes, the full form of RNA, students. Ribonucleic acid. RNA stands for ribonucleic acid. Now, remember that the DNA that we talk about, It is a nucleotide. It is a polymer of nucleotide. Now, what happens? You people know that there are nitrogenous bases present. Nitrogenous bases are purine and pyrimidines. We have discussed this. Now, these nitrogenous bases, when they combine with a pentose sugar, meaning a sugar that has 5 carbons in it, so a structure is formed which is known as nucleoside. Now, when a phosphate group is added to a nucleoside, then that becomes a nucleotide. And DNA that we have, it is the polymer of nucleotides. Now, in the nucleoside, the first one, wherein nitrogenous bases and pentose sugars are present. So in this case, the nitrogenous base is linked to the hydroxyl group, meaning the OH group of the first carbon of pentose sugar. Meaning, for example, all of you have studied the nomenclature of the compounds, right? You might have studied this in 11th also, and I think it's there in 12th. I don't know if it's there in 12th, but you have studied this in 11th, right? So we number the carbons. So carbon number 1, 2, 3, and 4 and 5. So now the nitrogenous base is linked onto the first carbon of the sugar through a bond that is known as N-glycosidic linkage. Okay, N-glycosidic linkage. Now see, normally what happens is that if these carbons are present, this first carbon already has a hydroxyl group present on it. Then on this hydroxyl group, the nitrogenous base comes and links to it, and the bond that is formed, because the linking requires a bond, and the bond that is formed is N-glycosidic linkage or N-glycosidic bond. Now, if we talk about the nucleotide, the phosphate group is an extra group that comes and attaches. And where does it attaches? So it attaches to the hydroxyl group of the 5 carbon of the nucleoside. So this position, the 5th carbon position. And the bond it forms is the phosphoester bond. So up until now, we've discussed about 2 bonds. The first is N-glycosidic linkage and the second is phosphoester bond. Now, who can tell me where does N-glycosidic linkage bond happens? Where is it? Like, where is this bond created? Where is it created? Tell me. It's right there on your screen, students. Read it and answer. Where is the N-glycosidic linkage formed? Tell me.
In the nucleoside.
Hmm?
In the nucleoside.
In the nucleoside. Where? So when the nitrogenous base, which is, it can be either pyrene or it can be purine. Pyrene is nothing. It can be either pyrimidine or purine, and it attaches to this particular first carbon of the pentose sugar with the bond that is N-glycosidic linkage. And then the phosphoester bond is present when the phosphate group is attaching. Now, when 2 nucleotides are linked together, now you have one nucleotide and then another nucleotide, and when both of them are linked together, so the bond that is coming between them is phosphodiester linkage. And this forms a dinucleotide. When 2 nucleotides are present together, a dinucleotide is formed. And then when many nucleotides are present together, so it forms a proper polynucleotide chain. A chain is formed. And this polynucleotide is what our genetic material is. Now, in a polynucleotide chain, phosphate moiety is present at the 5 end, because we know that while the nucleotide is being formed, the phosphate is present at which carbon? So it is present at which carbon? Tell me. What happened? You people are not understanding? Are you having any difficulty?
Ma'am, can you repeat the question?
The question is, the phosphate group attaches to which carbon number?
5.
5, yes, 5th carbon. So at the fifth position of a polynucleotide chain, there is the phosphate group. And at the third position of the sugar, we have the hydroxyl group present. So this is how a polynucleotide structure is there. Now, what is polynucleotide? When many nucleotides come together, they form a chain-like structure, and that is what we call as a polynucleotide. Now, this is a very important line. Many a times it is asked in the questions that backbone of the polynucleotide chain, it is formed by the sugar and the phosphate. Meaning it is like because of the sugar and the phosphate, that is why the polynucleotide has a strong or firm appearance. They form the backbone of it. Now, the nitrogenous bases linked to the sugar moiety, they project from the backbone. So, for example, if this is the backbone which is formed by the sugar and phosphates, and we know that nitrogenous bases are also present, so they're present like this. They project through it. Okay? Any doubts up until now?
No, ma'am.
Okay. Now we know that nucleic acids, they are of two types. There is DNA and then there is RNA. Now DNA and RNA, they are both the nucleic acid, but there is a difference between them. That difference is the sugar. The sugar in both of them are very different from each other. If we talk about the DNA, so what do we say? It's deoxyribose sugar. The sugar that is there is deoxyribose sugar, whereas in the case of RNA, there is normal ribose sugar. So in the case of RNA, every nucleotide has an additional OH group, which is present at the second position. And in RNA, we know that thymine is replaced by uracil. The thymine is not present in the case of RNA because it gets replaced by the uracil amino, like this thing, nitrogenous. So here, like this is the case, and always remember that the sugar in the case of RNA is a ribose sugar because it is ribonucleic acid. And the sugar in case of the DNA, it is deoxyribose sugar. Clear? Now, the thymine, like the chemical name of thymine is 5-methyl uracil. This is just an extra additional information. Now talking about the DNA. Now DNA, it is a very long polymer of deoxyribonucleotides. Meaning when many deoxyribonucleotides come together, they form a DNA molecule. And this DNA was first identified by this person whose name was Frederick Mesker in the year 1869. He was the one who identified the DNA. And then he named it as nuclein. He did not know what to name it at that point of time. So he named it as nuclein. So DNA is a long polymer. What is a polymer? You might have a chapter of polymer also in your chemistry. What is a polymer? A polymer is a substance which is made up of several smaller units that are monomers. Now, so the DNA was first identified by Frederick Mesker, and later on, many structures or many theories about DNA was made so as to find out what is the structure of DNA, what are the bonds that are present in the DNA, how is the DNA, the genetic material, and everything about it was studied. Now, in the year 1953, James Watson and Francis Crick, they proposed a double helix model for DNA. The DNA, the model for DNA that we talk about, like you might have seen that there is a helical structure that we see in the pictures of books and everything. That model, where did it come from? Who gave it? So Watson and Crick gave that model. And the name of that model is the DNA double helix model. Now, this research of Watson and Crick was based on the X-ray diffraction data which was produced by Morris Wilkins and Rosalind Franklin. Now, what happened in like before Watson and Crick, there were two people yet again, and they studied the DNA. They did an X-ray diffraction, and Watson and Crick based their model on the basis of the data that was collected. Any doubts up until now, students?
No, ma'am.
No, ma'am.
Everything is clear up until now? See, in this
Yes,
chapter,
sir.
maybe you will have a lot of doubts, and it's fine. Totally, you can ask all of your doubts. Okay, this chapter is a little out there. Now, talking about the next topic, that is Erwin Chargaff's rule. Now, you people know what purines and pyrimidines are? What are they? Collectively, if you talk about purines and pyrimidines, what are they? Tell me. Purines and pyrimidines are? Anyone? Please tell me what are purines and pyrimidines. Collectively, they are known as? Students, if you don't even know the answer, please try. At least say something. It's like I'm talking to myself.
Biomolecule.
Biomolecules. Okay, at least she tried. Anyone else? You can
Polymer,
say that they are names. Yes?
polymer,
Polymers. Okay. Some more imagination, please. Siddhika, you tell me what are purines and pyrimidines?
Don't know, ma'am.
They are nitrogenous bases. You people know what nitrogenous bases are, right? These are the nitrogenous bases. Now tell me the names of purines. Not studying at all. No one is studying at all. Okay, let's see. So Erwin Chargaff gave a rule, and this rule is applicable on double-stranded DNA. Now, according to him, purines and pyrimidines, they occur in equal amounts. Purines and pyrimidines, both of them are nitrogenous bases, and they occur in equal amounts. Now, purines of the DNA, they are adenine and guanine. A and G are purines, whereas pyrimidines of DNA are thymine and cytosine. What do you think will be present instead of thymine in the case of RNA? In RNA, we are talking right now about DNA. In RNA, what will be present instead of thymine? Student, this so I just taught. See here. Why are you people not paying attention?
This entire chapter is so confusing.
Yeah, it is. But if you pay attention, it won't be confusing. See here, in the RNA, thymine is replaced by uracil. It is a very big fact. All of you must remember that in RNA, thymine is not present. Instead,
Uracil.
uracil is present. Okay? Any doubts up until now?
No.
If you people don't have any doubts, then why are you not able to answer the questions? Tell me this.
We don't have a doubt. It's just that we're not able to recall it.
You're not able to recall it. Okay.
Yeah,
Preferably in the next class, you people will be able to recall it.
we'll see.
Can I hope that? Can I
Yes.
dream that? All right. Okay. Okay. Remember this, that a purine will always pair up with a pyrimidine and vice versa. So adenine pairs up with thymine and guanine pairs up with cytosine. What is the name of the bonds that are present in between them? What are these? Double bond, three
Triple,
bonds? What are?
triple bond. That is triple and that is double.
Both of the kinds of bonds that are there, these are the hydrogen bonds. Okay?
Oh, yeah.
Not double, not triple. These are the hydrogen bonds.
Hydrogen.
Now, A plus G, meaning adenine plus guanine, both of them are purines. And T plus C, both of them are pyrimidines. So their addition equals to 1. And this value is constant for all the species that have DNA as its genetic material. Now this ratio is fixed and the ratio is less than 1, meaning here we have written that it is equal to 1 and it will be less than 1 in the case of prokaryotes. For example, in the case of E. coli, it is 0.92. And the ratio is more than 1 in the case of eukaryotes, that is 1.52 in the case of humans. So this ratio tells us that the amount of purines and the amount of pyrimides will always be equal, exact equal. Any doubts? All right. Now let us talk about the features of double helix structure. But before that, you people note down till here, then we'll move forward. Also, now you don't have to be in a rush while you're noting down. Please read, please try to fit it in your brain and then you note down. You don't have to rush. You don't have to just quickly write down and I'm done. No. Read it, understand it, and then note it down. If you have any doubts, then ask me. You can switch off your videos and write comfortably.
Done, ma'am.
Everyone's done?
I'm 1 minute.
Everyone's done? All of you
Yes.
are done, students? Okay.
Yes, ma'am.
Ma'am,
Everyone's done?
Yes, ma'am.
Yes.
Done, ma'am.
Everyone's done?
Yes, ma'am.
All right, very good.
Yes, ma'am.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Done, ma'am.
Everyone's done?
1 second.
Okay.
Done, ma'am.
All right, very good. So now the next thing that we have to discuss is the features of the double helix structure of DNA. Now, this double helical structure of DNA, it was given by Watson and Crick, and these are the points that they mention. First of all, a DNA molecule, it consists of two polynucleotide chains. It is made up of two polynucleotide chains, meaning when several nucleotides come together, they form one polynucleotide. Same way, the DNA has two polynucleotide chains. Now, the backbone of the chain or DNA is the sugar and the phosphate group. Now, these two chains, they have a polarity. Now, the polarity is known as 5' and it is known as 3'. So, one polarity is from 5' to 3', and another polarity is from 3' to 5'. So they like both of these chains, like they are present in an anti-parallel fashion, meaning one will go from 5 to 3, so the other will go from 3 to 5. And remember that the nitrogenous bases, they are paired with the help of hydrogen bonds. And adenine will always form hydrogen bonds with thymine, two of the hydrogen bonds. Whereas guanine will always form hydrogen bonds with cytosine and three hydrogen bonds will be formed. And remember that the DNA that we have, it is a right-handed coil. Right-handed, meaning it is like this. This is right-handed and this is left-handed. So ours is right-handed helical structure. Now the pitch of the helix is 3.4 nanometer, like one turn is the pitch. And in one turn in the pitch, there are 10 base pairs present. Also, the distance between the base pair is 0.34 nanometers. Any doubts up until now, students?
No, ma'am.
Sure?
Yes.
All right. Now, how do we find out
Yes.
the length of the DNA? So, whenever you multiply the total number of base pairs